Hello, we want to look at the material at the end of Chapter 5. This is the material that was not covered in lecture on Wednesday, but yet some of this will be on the exam that we have on Friday. As we're going through this, pay particular attention to the material that is highlighted in red. Now, the first thing we need to look at are these terms exocytosis and endocytosis. And these two terms refer to a means of moving large molecules and particles into and out of the cell. So if you're moving something as large as a protein, you're going to have some means other than facilitated diffusion or the active transport that we've looked at so far. Because if what we've looked at so far, moving materials into and out of the cell, is either materials can slide in and out between the phospholipid molecules, or materials move through special transport proteins. Now, if you're talking about trying to get something as large as a protein, or even larger, into or out of the cell, you've got to have some means other than moving it through a protein. You've got to have a different means. So that's where these two come in. And exo means out of, cyto means cell, SIS on a word means a series of events or a process. So exocytosis, this is process of moving these large particles or large molecules out of the cell. Endocytosis, this is the means of moving these large particles or proteins into the cell. Okay, here we see a diagram from your textbook. So you notice in the diagram we have a vesicle that contains some kind of material inside the cell. Number two, this vesicle moves up here to the plasma membrane, fuses with the plasma membrane, and then number three, right here, the vesicle has fused with the plasma membrane, and that places its contents on the outside of the cell. Over here, we see an electron micrograph showing this idea. So here's vesicle, here's another vesicle, and the little black dots in there are some kind of particles contained in that vesicle. Right up here is a vesicle that has merged with the plasma membrane and is dumping its contents to the outside of the cell. So that is exocytosis. Down here on the measurements, you notice it's 0.25, and remember this little symbol right here, the lowercase scripted u, that is the Greek letter mu for micro, 1/1,000,000th of a meter. A micrometer is 1/1,000,000th of a meter. Okay, endocytosis. Endocytosis is means of moving these large particles into the cell. Large molecules or large particles into the cell. And we can subdivide endocytosis into these categories of phagocytosis and pinocytosis. Phagocytosis is the intake of solid particles, whereas pinocytosis is the intake of fluids and whatever happens to be dissolved in those fluids. Here's a diagram in your textbook showing how this would work. So we have some kind of material right here on the outside of the cell. The plasma membrane folds inward, and as it folds inward, that plasma membrane forms a vesicle around this material that pulls this material into the cell. And then what we see right here, this, and this, those would represent lysosomes containing digestive enzymes and acids. And down here on number 3, we see that these lysosomes have fused with this, what was now a food vacuole, and it's dumping those digestive enzymes and acids in there in the contents of that food vacuole, breaking it down. Okay, so also remember we had the terms primary lysosomes, that's the lysosomes before they have attached to a food vacuole, and then secondary lysosomes, that's the lysosomes after they've attached to a food vacuole. Now with endocytosis, We've got another form. We've got the phagocytosis, intake of solids, pinocytosis, intake of liquids. Now we have receptor-mediated endocytosis. And this is where specific molecules will attach to protein receptors on the plasma membrane. These specific molecules might be nutrients, or very often these are hormones. And these are then taken into the cell. Okay. Now we also have a term ligand, which is associated with this idea of receptor-mediated endocytosis. And a ligand is a molecule that bonds with a receptor protein. Okay. Then we also have the term coated pits. So you have a depression in the plasma membrane that contains the receptor molecules or receptor proteins. The interior of that membrane is going to be coated with proteins, and these proteins are going to activate those receptors to be able to accept the ligands. So if the protein coating on the inside of the membrane is not present, then the receptors do not have the correct shape and they cannot pick up the nutrient or the hormone from outside the cell. Okay, we have the term clathrin, also associated with the receptor-mediated endocytosis. So clathrin, this is the protein that coats the pits, the inside of the plasma membrane in these pits, and it activates those receptor proteins. So here's the diagram from your textbook showing this idea. And let's just start right here. So right here we have the receptor proteins on the surface of the plasma membrane. The gold would represent the plasma membrane. And coating this on the inside of the cell are these proteins called clathrin. Okay? So when the clathrin is attached to is coating the inside of that pit, then it causes these receptors to have the proper shape so that they can pick up some very specific material from outside the cell. In this case, what they're showing is that we have a low density lipoprotein particle. And so what these low density lipoproteins do is they carry cholesterol contained within this structure. So these are receptors for low density lipoproteins. So here the carrier for this low density lipoprotein, or this low density lipoprotein rather, can be pulled into the cell in the form of a vesicle. So this whole thing just pinches inward to form a vesicle. Now then, once this formed a vesicle, the clathrin proteins right here are lost from its surface, and these can be recycled to go back on other pits out here in the plasma membrane. Then, in this case, let's follow this through. We're going to have this acting like a food vacuole, and the lysosome is going to merge with that. So remember, the idea of primary lysosome is just the individual lysosome by itself. Here it has become a secondary lysosome, where the lysosome has fused with this food vacuole. Okay, and that's going to release the components of that low density lipoprotein particle out into the cytoplasm. Let's go back up here. Here's the portion of the plasma membrane that contains the receptors. This portion will pinch off and forms its own vesicle, and then this will fuse with the plasma membrane and once again place those receptors on the surface of the cell. Then once that pit is coated with clathrin inside, those receptors are once again able to act as receptors, and we're back to our original starting place. Okay, here is this concept shown with the electron micrograph. And we can see, if I can find the cursor, there it is. Here we can see the dark line is the plasma membrane. Okay, this right in here to the inside, that's the coating, protein coating, the clathrin. And then this would be the, these would be the receptor proteins. Okay, so we have that stage. Then this folds inward more. Then here it is just about to pinch off and form a vesicle. And here this has formed a vesicle and it still has that clathrin protein on the surface.